quiz back, I can give it to you at the break. Okay, so what we're going to focus the first part of the class today on the Davenport diagram, which is basically it's a two-dimensional graph, but it's a three-dimensional. It has informations in 3 dimensions of the Henderson-Hasselbalch equation for physiological pH buffering. That is to say, the buffer system of carbon dioxide and bicarbonate. We'll talk about some more information about partial pressure. And actually, I stopped updating this. We'll also talk about the mechanisms for gas transport by blood. Okay? So here is the Henderson-Hasselbalch. This is the Davenport diagram. And it looks confusing, and you have a copy of it. And so here is the Henderson-Hasselbalch equation, which you don't have to know. This is the pKa of the bicarb carbon dioxide system. This is the graph. It's hard. It looks superimposing, and so what I'm going to do is build it in front of you. And so what I'm going to do is just draw the graph. And then blanket. Okay? So this is the Davenport diagram. We're plotting blood pH on the x-axis, and this is blood concentration of bicarbonate ion on the y-axis. And so pH, the normal pH, which I am going to ask you to know, is 7.4. The normal bicarbonate concentration is 26, which I don't care that you know that, but knowing that it's between 20 and 30 is important. The normal partial pressure, whoa. The normal partial pressure, I guess we're talking about arterial partial pressure of CO2, is 40 millimeters of mercury. That's a normal value. I'm going to get hurt. So, remember the Henderson-Hasselbalch equation in this setting was pH is equal to pKa, what is it? Somebody help me out. It's plus the logarithm of the concentration of bicarbonate, right? Isn't that up here? Divided by, well, it's the solubility of CO2 times the PaCO2. Is that right? Okay. So there are three. The solubility of CO2 is a constant. We're in water, and we're just talking, or in serum. So, but there are three variables. pH. Well, I mean, there are three variables that define pH. There are three variables. pKa is a constant. That's defined by the chemistry of carbon dioxide and carbonic acid and bicarbonate. That's chemistry. Solubility is chemistry, so that's a constant. But there are three variables here. There's pH, there's the bicarbonate concentration, and there's the carbon dioxide partial pressure. So we have three knobs that we can turn, three things that we can change, and only two axes on the graph. So that makes it a little tricky. But we simplify it by realizing that, okay, for a given, if we just say, okay, let's take a given PCO2. Let's just take a normal value, 40. Well, then this becomes a constant. We just say, okay, we're going to set PCO2 at 40, and now we have a relationship between pH and bicarbonate. And that's what we're plotting, a relationship between pH and bicarbonate. Now, you would say, well, this is weird. Shouldn't pH be on the y-axis as the dependent variable? You've put it on the left of the equation, and shouldn't bicarbonate be on the x-axis as the independent variable? And if it were me creating if my name were Davenport and I was creating this figure, I would have done it that way. But I'm not Davenport, and he didn't do it that way. He did it this way. And so this is the way we're going to learn it. But it doesn't really matter which is dependent on which. They're codependent. They're dependent on each other. And so the graph plots bicarbonate concentration on the y-axis and pH on the x-axis. If you want to convert it into the other way, that's fine, but then you have to teach me what you did. It's not worth it. So we have a normal point. The normal PaCO2 is 40. The normal bicarbonate concentration in plasma is 26. The normal pH is 7.4. That's a normal point. That point lies on a curve that is defined by that partial pressure of CO2. So if we define the partial pressure of CO2 at 40, we are basically saying everything, in order to agree with the Henderson-Hasselbalch equation, all the points have to be on this curve. All points on this curve are the representation of the Henderson-Hasselbalch equation for a PaCO2 of 40. Now remember, in chemistry, the weak acid and the weak base and the strong acid come together. And so you don't do this in chemistry, but in biology and physiology, the kidney, the control of bicarbonate, we can independently control bicarbonate through the kidney. We have multiple ways to control bicarbonate, and we have multiple ways to control pH. And we have multiple ways of controlling PaCO2. So the point is, you don't do this in chemistry because you have less control for any given acid-base system. So this is the curve that represents the Henderson-Hasselbalch equation for a given normal PCO2 of 40. So now let's look at this. If the pH goes down, that is to say we're becoming acidotic, the bicarbonate concentration goes down. Okay, well let's do it the other way. If the bicarbonate concentration goes down, then by just plugging in a lower number here, we're going to get a lower number here, and that's acidosis. So there's a relationship between acidosis and low bicarbonate at a given PCO2. And similarly, if bicarbonate goes up, if the kidneys start holding on to bicarbonate, then this ratio goes up and pH goes up, and we become alkalotic and high bicarbonate. Okay? So far, so good. This curve represents the Henderson-Hasselbalch at a given respiratory condition with a normal PCO2. So there's another curve right here. Remember, this is a surface, a two-dimensional surface. There's another curve right there that represents PCO2 of 39. And there's another curve right here that represents PCO2 of 41 millimeters of mercury. Right? There's a whole series of curves that define a two-dimensional space. Each of those curves represents a different PCO2. So the Davenport diagram that you have shows 3 sample Henderson-Hasselbalch equations. One with very low PCO2, like 20, is that the one you have? And one with very high PCO2 of 60. But there's a whole family of curves in between 40 and 20 and in between 40 and 60. Okay, that's the thing to realize. So I'm going to use the example that is in your handout. Let's go to a curve that is out here where the PaCO2 is 20. Isn't that right for in the curve? The one down here is 20 millimeters of mercury. Is this hypo or hyperventilation? You have it in the handout, but you should understand that if we have a PaCO2 of 20, we are hyperventilating. We're still on a Henderson-Hasselbalch curve, but we're now at an abnormal partial pressure of CO2. We have hyperventilated, we have blown off more CO2 than our body wants us to blow off. This is not the exercise setting. This is hyperventilation. So moving from this curve, the blue curve, to the red curve is moving from a normal ventilation to hyperventilation. And if we are in a hyperventilating state, we are on this new curve. Now, if your brain detects, so we're moving onto this curve, and we are becoming alkalotic. We have alkalosis. Our blood is too alkaline. Chemoreceptors detect that pH, that high pH, that alkaline pH. Our body says, the system says, okay, you're hyperventilating. The most important thing that we need to do is make sure that the pH stays in the normal range, pH 7.4, very close to pH 7.4. So we're on this new curve. You're hyperventilating. What can we do? Our body, can lower bicarbonate. The kidneys can excrete bicarbonate in order to normalize the pH. So this is hyperventilation. This arrow represents hyperventilation, moving from the normal curve to the hyperventilation curve. And these arrows represent metabolic, what do I use in the word, I mean in the figure? Metabolic compensation. Metabolic being something other than the respiratory system. In this case, it's renal compensation. But for now, let's just call it metabolic compensation. It's renal compensation. Okay, so we've clarified some of the Davenport diagram. Let's go to a different condition. I want a different color, and the orange isn't working. I've already used blue. Okay, I can use black. Okay, so like I said, there's a whole family of curves. There's a whole family of curves that are up here, right? So remember, there's 39, 38, 37, 36, 35, down to 20 here. And now there's 41, 42, 43, 44, 45, all the way out to, what do I use in the example? 60? Yeah. So 60 millimeters of mercury. High partial pressure of CO2 means we are hypoventilating. And if we started normal and we started holding our breath or exercising without breathing more, we would be hypoventilating and we would move. So this is the curve that we follow when we hypoventilate. So this is hypo, this arrow represents hypoventilation. Now, what do we do? So using the example from hyperventilation and renal compensation, what do we do to, we're now becoming acidotic. The pH is deviating from our normal 7.4 into the acid range. What happened? What do the kidneys do? Say what? Go ahead. Somebody? They what? They hold on to bicarbonate. So we increase the serum bicarbonate in order to have a pH that is close to normal. We're not on the normal curve because we're hypoventilating, but we are at a normal pH. And so this is hypoventilation. Now this arrow represents hypoventilation, pretty severe. And these arrows represent, let's just call it renal compensation. In order to normalize pH, given a higher than normal PCO2. So what you're seeing is that if CO2 goes up, the body tries to make the bicarbonate go up so that the pH doesn't change. And if the PCO2 goes down, you go back to the equation. If the PCO2 goes up, the body tries to increase bicarbonate concentration so that this ratio stays relatively normal and the pH stays normal. If the PCO2 goes up during hypoventilation, then the body tries to increase bicarbonate so that this ratio stays normal and the pH stays normal. So we have mechanisms for trying to control this ratio. Okay, let's do another example. Everybody with me so far? So what we're talking about is a respiratory abnormality, hyperventilation or hypoventilation. That's a respiratory abnormality. and a renal compensation. Let's now flip this. Let's talk about a metabolic abnormality and a respiratory compensation. So let's do a situation where the body is becoming acidotic. Not because the PCO2 is still normal. We're breathing normal, or we're breathing normal, but not enough. Well, you'll see. So what are some ways that the body can become acid that don't require a change in the respiratory system? Well, if the kidneys start excreting more bicarbonate than they should, that's an easy one. Or if you are exercising and starting to generate lactic acid, you will generate acid and your body will become acidotic. So those are two simple ways where we can imagine the body getting acidotic. We're still on the Henderson-Hasselbalch equation. We're still on the normal curve because we haven't adjusted respiration yet. Okay? So this we would call, this arrow right here is metabolic, which just means anything other than respiratory, in this case. It could be renal, it could be muscle. Metabolic acidosis. And what's the respiratory response? Do we go to this curve? Or do we go to this curve? Do we hypoventilate, or do we, no, I shouldn't use that word. Do we decrease ventilation, or do we increase ventilation? Increase it, because by increasing respiration, we can get to a pH that is closer to normal. And so this would be a respiratory compensation to a metabolic abnormality. I'm trying to use different kinds of arrows. So the solid arrows represent the abnormality, and the dashed arrows represent the compensation. So your figure doesn't do that, but you can decorate your figure to help you with that. Okay? So let's just do one more. Well, so this is an example, right? Let's say what's going on here, let's say that we're exercising and we're generating lactic acid because we're not breathing enough. We're becoming anaerobic. We're generating lactic acid, so we want to ventilate more. Bicarbonate is going down, so we want to get rid of CO2. Bicarbonate is going down because we are becoming acidotic. So we want to decrease the partial pressure of CO2 in order to try to renormalize pH. Acidotic because of exercise, increase ventilation as a response. You do this almost every day. Well, every day that you exercise. Okay, so let's do another case. Let's go to metabolic alkalosis. Something is changing in the body. The kidneys are holding on to more bicarbonate than they should. We are excreting more acid than we should, other than respiratory. So this arrow represents metabolic alkalosis. Something is pushing, we're still breathing on the normal curve. Our PCO2 is still 40. But something else is pushing us away from normal. We haven't talked about renal mechanisms, so let's not talk about that. But can you think of a way where your body would be excreting more acid than it should? You've all experienced it. Say again?
Through the kidneys, the uric acid?
Let's not talk about kidneys, because that's complicated. We're in the I'll start to give you some hints. We're in the digestive tract now, and we're excreting acid. I'm sorry, say it louder. Heartburn. Well, not really heartburn, but you're close. Vomiting. That is large-scale excretion of acid, right? Because you're vomiting stomach acid. So chronic vomiting is, in fact, producing a metabolic alkalosis. You are excreting a bunch of acid. We haven't talked about the digestive tract yet either, but you'll learn about gastric acid pretty soon. And so vomiting a bunch of acid is a form of excretion of a bunch of acid, and so this is metabolic alkalosis. So what does our body do to respond to metabolic alkalosis? Well, we do respiratory compensation for the metabolic alkalosis to try to keep the pH normal even though we're excreting a bunch of acid, and this is respiratory compensation for metabolic alkalosis. You're probably not aware of it, but if you are in a condition of chronic vomiting, you're gonna breathe. You breathe shallow, and you breathe slower. Now you feel pretty bad, so you're not aware of your breathing. That's the least of your problems right now, but that's what your body is doing. Okay, so there's the Davenport diagram. We've created almost all of it, and those are the important arrows. So now it doesn't look any simpler than this. Okay, now it doesn't look any simpler. I'm going to go ahead and erase, unless there's an arrow that you want me to clarify. Jasmine.
So for metabolic, respiratory response to a metabolic abnormality, the PaCO2 doesn't change at all.
Oh, it does. The respiratory compensation to a metabolic alkalosis is to go to a PCO2 curve that is lower. So we're moving to a different CO2 curve. Yeah, PCO2 is changing. It's going down so CO2 is going down in order to compensate for the decrease in bicarbonate. Yeah, so when we move from one curve to another, whether it's a compensatory change or whether it's an abnormality change, we are going from this curve to this curve is changing PCO2. Are there other questions? Yeah, Zoe.
Can you explain why increasing ventilation decreases the PaCO2?
Increasing ventilation. So you mean why when we hyperventilate? When we hyperventilate, why do we decrease PCO2? Yeah. Okay, so we are ventilating the alveoli so much that CO2 from our body is being excreted through exhaled breath more than it should be. So in normal ventilation, there's normal equilibration of the blood with the alveoli, and the alveolar PCO2 is 40, and because of the perfect diffusion of CO2, the arterial PCO2 is 40. But if we hyperventilate, we're now making our alveoli closer to atmospheric, that is to say closer to zero, and because of the perfect exchange, our arterial PCO2 becomes closer to zero. Well, it never goes to zero or else we're dead, but it can go as low as 30 or 20. These are good questions, other questions like this. Because if you can walk through the Davenport diagram pretty much the way I just did, so I'm going to go ahead and put the whole thing up and erase. so that we can see it. I'm going to make it. I'm going to do this without hurting myself. What's that? There's a chair. Oh, yeah. Okay, now it's too late. Yeah. So there's the whole Davenport diagram. And I think we've covered all of the arrows. And the point is, deviations are compensated in order to normalize pH. Deviations are compensated in order to normalize pH. pH is the thing that we need to control most. Because our enzymes, our whole body is conditioned, has evolved to work best at this pH. Okay, so any questions? It requires some work. There's a problem in your problem set that's actually pretty straightforward that asks you to use the Davenport diagram. It will be on an exam. It will be on more than one exam because when we get to the kidney and when we get to the digestive tract, I might come back to the respiratory system in order to, flesh that out a little bit more. So we've talked about some of these things. Production and buffering of acid. So when you eat, when you digest, you produce acid. Your lungs control CO2. Your respiratory system controls the arterial concentration of CO2. But your kidneys also control, they don't control CO2, but they control acid secretion, excretion we call it in the kidneys, or acid retention. So your kidneys, by controlling bicarbonate excretion and acid excretion, participate in this control of pH. So the respiratory system and the renal system are the two have the biggest knobs, and but the digestive system and the muscle system also have some control. So let's just think about respiratory. Remember now, the name here is if it's respiratory acidosis, it means that the primary defect, the primary abnormality is respiratory. So we're talking about hypoventilation. Bless you. So there's some disease settings like asthma and emphysema where you don't ventilate enough. Hypoventilation. And we'll talk about some, we have a respiratory case study. So we'll talk about a respiratory case this afternoon. Respiratory alkalosis, hyperventilation. Well, anxiety can cause hyperventilation. If you're at high altitude and you're hyperventilating because you have oxygen hunger, that can lead to, when you go to 12,000, 15,000 feet, your need for oxygen starts to overpower your need for your body to control CO2, and you in fact hyperventilate until your body readjusts to the higher altitude. So altitude sickness is a semi-normal response to a rapid change in altitude. We weren't evolved to fly from sea level to 14,000 feet. In fact, we weren't evolved to walk from sea level to 14,000 feet. If we are going to live at 14,000 feet, we were evolved to move there slowly and adjust. Okay, and so metabolic acidosis is a metabolic reason for a low pH. And anaerobic metabolism, that is to say, exercising but not breathing enough. Our muscles are becoming anaerobic. Kidney disease, excreting, retaining too much acid or excreting too much bicarbonate. Severe diarrhea actually is the secretion of alkali. Metabolic alkalosis, well, for example, we talked about a vomiting situation that would generate metabolic, that is to say, not respiratory, alkalosis. So here are some common examples that can produce respiratory abnormality or metabolic abnormality that affect pH. Quoi?
Why is diarrhea?
We'll get to that when we get to the digestive tract. Let's not talk about that yet. Let's focus on breathing. Okay, clicker time. This is before any compensation. So if you jump to 14,000 feet and before compensation, what happens? We're just talking about the respiratory defect. Right, B. It increases because you're hyperventilating. Hyperventilate, increase. Okay, what happens to blood pH during an anxiety attack? It increases. Same idea. Your hyperventilation is one of the hallmarks of an anxiety attack. Okay? What happens to blood pH during exercise? And now I'm thinking about both the event and the compensation. Okay, I'm seeing best answer, I'm seeing some mixed here, so maybe we'll spend some time. It decreases, but only a little is the best answer. So let's just see if we can understand that using the Davenport diagram. Exercise is metabolic acidosis, so it decreases, but then we compensate, and you can say, well, okay, it should stay the same. But was stay the same one of your answers? Well, that wasn't one of the options that I gave you. So it has to be on the decrease side, and it's only a little because of compensation. Does that make sense? Did I? It stays the same. Okay, so A could be an acceptable answer to that. A and C are both acceptable answers. It doesn't increase. pH doesn't increase. Proton concentration increases, but pH decreases. Make sure you keep track of the fact that pH and the amount of acid are inversely proportional or inversely related. And you have to read my questions carefully. Sometimes I'll ask you the concentration of acid or protons or the amount of acid, and sometimes I'll ask you about pH, and you have to translate back and forth. Low pH is high acid. You just have to remember, and you just have to remember. Okay, one of the cures for hiccups is to breathe into a paper bag, re-breathing expired gas. So, breathing into a bag, rebreathing the gases that you exhaled, rebreathing. What happens to blood pH? Okay, I'll put up the Davenport diagram. What happens to blood pH? Is we are rebreathing expired gas. Is PCO2 increasing or decreasing? Increasing. We're breathing an atmosphere that is now high in CO2 because we're breathing the stuff that we exhaled. So we're breathing a high CO2. PCO2 increases. So what happens to pH? Decreases. Okay, so you can see that the Davenport diagram really helps you answer questions that I could ask you. Questions about this? Let's do this and then take a break. So this is a figure from your textbook, and it makes it more complicated than it needs to be. The main point is that the smooth muscle that controls airway and therefore controls ventilation, and the smooth muscle that controls arterioles in the pulmonary circulation and therefore controls perfusion of the alveoli is normally matched. So if there is a region where airflow to a part of the lung is decreased, some kind of bronchoconstriction, then we constrict the blood flow to those areas to try to match perfusion and ventilation. And how do we do that? Well, you know, we can, arteriolar smooth muscle can contract. It's probably a GQ coupled story. If there is a region where blood flow to the lung is impaired, maybe due to an injury or an anatomical defect or a clot, a pulmonary clot, a region of the lung that is not getting perfused, there are mechanisms to constrict bronchial smooth muscle, GQ, to try to limit airflow to that region so that ventilation and perfusion is matched. It's a phenomenon that happens. And so the idea is that blood is diverted away from the areas where it's not going to be useful and to the areas where the alveoli are well ventilated in both cases. Ventilation, perfusion, matching. We're going to see that in lab this afternoon, actually. So let's take a break, and then we'll come to an equation that we are going to use in lab today. So I want to teach it. And then we'll start with the transport of oxygen and CO2 by blood. That's where we're going. The alveolar oxygen equation. And I think it's kind of interesting. And we'll use it in lab today. So it's very interesting. So remember, capital A represents alveolus, alveolar, and little lowercase a represents arterial. So alveolar oxygen is equal to the inspired oxygen, the partial pressure of inhaled or inspired oxygen, minus the alveolar partial pressure of CO2, which is equal to the arterial partial pressure of CO2, divided by this thing called the respiratory quotient. Okay, so this is, where does this equation actually makes some sense from metabolism and from physiology. So what is in our alveoli, in terms of partial pressure, is what we inhale minus the CO2 that we produce. Because CO2 is taking up partial pressure. And the R comes from the digestive system. So here's an interesting equation that you know. This is not yet balanced, but this is complete oxidation of glucose. This is what they're learning down the hall in Self-Phys right now. Some of your friends might be struggling with this as we speak. Let's balance it. Some of you may already have memorized the numbers that go in front of these. How many oxygens? Pardon? Okay, we can do it. How many carbon dioxides? 6. We got a balanced carbon. So 6 carbons, 6 carbons. Somebody else tell me a number. For what? For water. 6 waters. 12 hydrogens? 12 hydrogens. So how many oxygens? 6. So this is 6, 8. Wait, I did something wrong here. No. 6, O2. That can't be right. No, but it's got to be 6. So something's wrong here. Is this right?
18 oxygens on both sides.
Oh, that's right. There's oxygen here. Right. There's 18 oxygens here. There's 18 oxygens here. 6 O2s. I knew this was 6. I just was getting confused by the math. Okay. What's the respiratory quotient if we eat a pure carbohydrate diet? We produce exactly as much CO2 as we consume in oxygen. The respiratory quotient for pure carbs, if we eat nothing but carbohydrate, the respiratory quotient is 1. We have to exhale exactly as much CO2 as we inhale in order to stay in balance. So the respiratory quotient for pure carbohydrate is 1. The respiratory quotient for if we eat nothing but fat, the respiratory, you can Google the complete oxidation of a fat, and we produce less carbon dioxide than we consume in oxygen. There's a larger number here than there is here for the complete oxidation of a fat, and the respiratory quotient is 0.7 for pure fat diet. And for pure protein diet, it's somewhere between 0.8 and 0.9. So I just chose 0.85. But the point is, we don't eat pure carbohydrate, we don't eat pure fat, I hope, and we don't eat pure protein. We eat a mixed diet. Even if you're on one of those special Atkins diet, you're eating something that's mixed. And the typical R is, I think I put it up there. Typically, it's 0.8. for a mixed diet. And all the clinicians, if a clinician is calculating the alveolar oxygen equation, or even if a machine is doing it, they're just assuming a mixed diet. And, you know, a mixed diet can vary, but it doesn't vary by much. And so the respiratory quotient is used is 0.8. And you will use 0.8 in lab today. So here's an equation. So I wanted to introduce the equation, and now I'm going to just move ahead because you'll use it in lab today. The other concept that you will use in lab today is that remember, oxygen didn't equilibrate perfectly across from alveoli to blood, right? So if we talk about PaCO2 and we look at the difference between alveolar and arterial CO2, what's the difference normally between arterial and alveolar CO2? 0. That difference is usually 0. The PaO2 and the PaO2, which is often called the AA difference, is not 0. It's somewhere between 5 and 15 millimeters of mercury. And you'll use that information in lab today. We can measure arterial oxygen partial pressure because we can do blood tests for arterial oxygen. We can calculate alveolar oxygen by knowing the inspired oxygen and measuring arterial CO2 and assuming a normal R.
What did you say that is called?
The AA difference. It's the difference between alveolar and arterial O2. Okay, we'll play with this in lab today in a case study. So you'll be doing some diagnosis, and you'll be using some of the tools that diagnosticians develop when they're talking about a respiratory abnormality. So let's switch gears now and talk about how gases are transported by blood. I mean, you know, this is an important part of the respiratory system, right? The gases have to be exchanged in blood, and then the blood has to deliver gases or remove gases, deliver oxygen and remove CO2, and that's what the job of blood. And so let's talk about that mechanism. So you probably already know that the main thing that carries oxygen in the blood is hemoglobin, and we're not going to get into the biochemistry of hemoglobin too much because I think you do it in biochem. The point is that hemoglobin has a heme group. This is the structure of a heme group. Globin is the protein. Heme is a chemical group that is attached to globin to make hemoglobin. So there's a globin polypeptide. It's got a nitrogen, I think an asparagine, and the asparagine nitrogen is covalently bonded to this heme group. And the heme group has iron in the core of it. And you know that iron is a necessary nutrient because our blood needs iron in order to make heme, in order to carry oxygen. That's why we have to have a certain amount of iron in our diet. So this is the bond, and this is that oxygen interacts with that iron in the center of hemoglobin. So I'm not going to ask you to memorize or know much about the structure of heme or globin. It's just, you know it's there. Hemoglobin binds oxygen. How many of you have seen the oxygen saturation curve of hemoglobin already? Maybe even this year. Okay, so some, but not everybody. Okay, so let's just walk through this. This is looking at the partial pressure of oxygen in a solution. No, in blood. So partial pressure of oxygen from 0 to 100 millimeters of mercury. And this is plotting the percent saturation of hemoglobin. So if there's no oxygen is bound to hemoglobin and the percent saturation is 0. If there is 30 millimeters of mercury of oxygen, about half, well, let's see, 26 or whatever. It's not a number to know. I'm just using the curve. At a relatively low partial pressure of oxygen, only about 25, hemoglobin is 50% saturated. We're dead down here, by the way. At least if this is arterial partial pressure of oxygen. So oxygen, there's two ways that oxygen can be carried in blood. It can be dissolved in the serum, and that's a tiny minor component, or it can be bound to hemoglobin, and that's the major component. The dissolved amount for oxygen is trivial, but I introduce it because for carbon dioxide, it's not trivial. Okay? But this is just oxygen dissolved in water, in serum. This is oxygen bound to hemoglobin. So at a normal arterial PO2, we are we're about 95% of the hemoglobin is saturated. Or 95% of the sites of hemoglobin are occupied by oxygen. And the brown curve is just what we add if we added the yellow line to the red curve, right? This is just this amount here added to this amount here. And we get to 97.5 total oxygen carrying capacity of blood. So when the blood leaves the lungs, it is 97% of the hemoglobin is saturated, is bound to oxygen. And when the blood re-enters, the superior and inferior vena cava and is about to get transported to the blood, this is now venous blood that has been depleted of oxygen. It's not depleted all the way. It's depleted to about 40 millimeters of mercury of partial pressure of oxygen. And the saturation is around 75% at that concentration. So this is the operational range of hemoglobin in our blood. Venous, arterial. Venous, arterial. We're almost never down here. Now, in biochemistry, I'll bet you learn about, what's the word I'm searching for? Cooperativity. Yep. You learn about the fact that this curve is sigmoidal, S-shaped, and very steep, is a diagnostic of the cooperativity, the fact that there are 4 subunits of globin per hemoglobin, and there is positive cooperativity to the binding of oxygen to hemoglobin. So I've mentioned cooperativity so that I am in agreement with, who's teaching biochem this year? Kalani? Yeah. But I'm not going to go into the details of cooperativity or cooperative binding. Hemoglobin is the poster child for cooperative binding. Okay, so now let's walk through the carrying of oxygen by blood. And this is a figure from your textbook. And but it's an okay figure. So, but let's, and I gave this to you big enough, I think, to label. So each of these arrows represents a function, represents an action. In other words, it has a verb associated. It has a concept associated with it. So what is the word that we would use to describe how oxygen gets from air into alveoli? There's a word that describes this arrow. Ventilation. This is ventilation. Say what? Ventilation is bringing oxygen in and getting carbon dioxide out. So we're only looking at the ventilation of oxygen right now. So this is ventilation of alveoli. There's a word for how this oxygen gets from alveoli into the plasma of the blood that is perfusing the alveoli. Remember the cell types we're not seeing them, but there's type 1 pneumocyte, type 1 alveolar cells here that make the thin barrier. We don't see them, they're so thin. There's type 2 that make the surfactant, which we don't see because it's a molecular layer. Remember, don't forget the cells, but they're there. What's the name of this arrow? Diffusion. So oxygen diffuses into blood. It diffuses into blood plasma, where some of it, a very small amount of it, is dissolved. What's the name for the arrow that moves oxygen from the blood plasma into a erythrocyte, into red blood cell? Diffusion. What's the name of this arrow that is showing an equilibrium? It's actually an equilibrium between hemoglobin and dissolved oxygen and hemoglobin that is coordinated with an oxygen. There's a word for this arrow. Binding. Oxygen is now bound to a red blood cell. No, it's bound to hemoglobin inside a red blood cell. What's the name of the word that takes blood from a lung capillary and delivers it to a tissue? Say what? Transport, that's a good word. Blood flow. I mean, blood moves from one place to another. That's what your circulatory system did. And so we're moving blood, including the red blood cell, from the lung capillary to a tissue capillary. Blood flow. Okay, so what's the word for hemoglobin letting go of oxygen and it becoming, what's the word for this arrow? Unbinding or dissociation. So now everything is going to be reversed. What's the name for the arrow that allows unbound oxygen to move from the cytoplasm of a red blood cell to the serum, the blood plasma outside of the red blood cell? Diffusion. What's the name for this arrow? Diffusion. More diffusion. Lots more diffusion. What are these things? Mitochondria. What does the mitochondria do with the oxygen that has diffused to them? What comes out of this arrow? Water. Electrons and hydrogen atoms, ions, which we don't follow in self-is. Well, we follow the electrons, that's for sure. We don't follow the protons. But the electrons, oxygen is the final electron transport acceptor. Where did those electrons come from? Two words. One begins with an N and the other begins with an F. NADH. FADH2. The high energy electron carriers. It all comes back if you let it. The electrons and oxygen combine to form water. And that's why the oxygen is diffusing to the mitochondria. This is pretty easy. Any questions about oxygen transport? Yeah.
For the dissociation of oxygen, in the
This one here?
over there, yeah, is that also an equilibrium, or is that a one-way?
Well, it's moving. It's tending to move in this direction because.
I'm talking about within the red box.
Oh, that's everything is unequal. All of these arrows are bidirectional, but if this arrow is pulling oxygen off here, then this arrow is more in this direction. So all of these arrows are in fact reversible. All of them. But the arrows here are showing the net movement in the lungs and in the tissue. Okay, so oxygen binds in the lungs and unbinds in the tissues. There are some features of hemoglobin that assist this. And I don't want to get into the great gory details. I think I gave you one of these graphs. The main point is, here is the hemoglobin binding curve at 37 degrees, normal body temperature. When your body temperature is high, like during fever, or during exercise, or in a location that's high, like a muscle that is exercising strenuously, it's on a slightly different curve, and we move, instead of moving to here, we move to here. In other words, we unbind more oxygen at high temperatures. And that's physiological in order to provide more oxygen to high temperature muscles while exercising. This is exaggerated. 41 degrees is too high, but it's just like PaCO2 of 20 is too low. It's trying to show you, there's another curve here that is at 38 degrees and 39 degrees and 40 degrees. There's a whole series of curves here, and this is showing an extreme example. So temperature, heat, helps hemoglobin let go of its oxygen. And carbon dioxide, so here's the normal curve, 40 millimeters of mercury. Carbon dioxide also helps hemoglobin let go of its oxygen. And where is carbon dioxide high? In your muscles during exercise, because that's where metabolism is happening. And acid, here's normal, here's acidic pH. Acid also helps hemoglobin let go, unbind its oxygen. So high temperature, high CO2, and high acid, low pH, help hemoglobin let go of its oxygen so that it can be delivered to those tissues that are hot, like muscle, rich in carbon dioxide, like muscle, low in acid, like muscle, especially during exertional exercise. So when you exercise and you are working out, your muscles help your blood let go of the oxygen that the muscles need. It's pretty interesting. Okay, so I'm not going to ask you to know how to draw these figures. I do want you to know that hemoglobin has to let go of its oxygen. The oxygen has to dissociate from hemoglobin in the metabolically active tissues. And in fact, metabolism, heat, carbon dioxide, and acid, some of the products of metabolism, assist in the blood delivery of oxygen. That is to say, allows the hemoglobin to dissociate more oxygen than it would have otherwise. It's sort of a beautiful system. Hemoglobin's a pretty good molecule. All right, so what I want to do now, so this is the figure, and I gave you this one as well on a large scale, right? So let's do this for a few minutes. I'm not going to hold you long or over, but I want to start this. And then we'll pick this up on Tuesday. You don't need this for the problem set, and you don't need it for lab today. But I do want to use this time. So here we are in the tissues. We're starting in the tissues now because we're producing CO2. What are the parts of metabolism that produce CO2? The self is Krebs cycle, pyruvate oxidation. Oxidative phosphorylation doesn't, but pyruvate oxidation and Krebs cycle and beta oxidation of fats, which you didn't learn about in self-is, all produce CO2. So this is CO2 production in your metabolically active tissue, muscle, brain, liver, whatever. What's the name of this arrow? Let's just do, maybe we'll do half this. What's the name of this arrow? Diffusion. What's the name of this arrow? That's diffusion. What is this CO2 doing here in serum? It's just dissolved. This is dissolved CO2. Remember, the partial pressure of CO2 in blood is 40 millimeters of mercury. That partial pressure of CO2 came from the solubility of CO2 times the partial pressure, which generated an amount or a concentration of dissolved CO2. There's some of the CO2 is dissolved. Okay, so now so this arrow is just diffusion as well. Because it's dissolving from the cells into the blood plasma. This arrow here, so let's just finish this arrow here. What's that arrow? Well, that's also diffusion. So I'm just going to put D here for diffusion. Some of that CO2, so now we're inside the erythrocyte. We're in the cytoplasm of the red blood cell. Some of that is now dissolved in the cytoplasm. It's just dissolved CO2. Not a large amount, but it's what contributes to the partial pressure of CO2, so it's not trivial. Okay, so this is diffusion. What is this arrow? And what is Ca? This is the enzyme carbonic anhydrase. That catalyzes or accelerates the reaction that allows the CO2 that just diffused there and the water that's there, because it's a fluid, to become carbonic acid. This is the chemical association of CO2 and water. And let's stop there. I would like you, maybe as homework between now and Tuesday, in addition to working on your problem set, remember the problem set's due on Monday, but if you can come to class prepared, we'll be able to go through this relatively complicated slide more quickly on Tuesday.